What is a heterotroph? Let's explore this fundamental concept in biology. Heterotrophs are organisms that cannot produce their own food. This is their defining characteristic. To understand heterotrophs better, let's compare them with autotrophs. Heterotrophs cannot produce their own food, must consume organic matter, and obtain energy from other organisms. In contrast, autotrophs can produce their own food using sunlight or chemicals, making them self-sufficient for energy. Examples of heterotrophs include animals, fungi, and many bacteria, while autotrophs include plants, algae, and some bacteria like cyanobacteria. Heterotrophs play crucial roles in food chains and energy flow throughout ecosystems. Energy flows from producers, which are autotrophs like plants, to primary consumers, which are heterotrophs like herbivores. Then to secondary consumers like carnivores, which are also heterotrophs. Finally, decomposers, another type of heterotroph, break down dead organisms and return nutrients to the soil. Let's summarize the key points about how heterotrophs acquire energy and nutrients. Unlike autotrophs, heterotrophs cannot convert sunlight or inorganic sources into energy. They must rely on breaking down complex organic compounds obtained from other organisms. They obtain carbon from organic sources and serve as essential links in the flow of energy through ecosystems. Holozoic nutrition is a form of heterotrophic nutrition where organisms ingest solid food particles and digest them internally. Unlike autotrophs, heterotrophs that use holozoic nutrition cannot synthesize their own food and must consume other organisms. Holozoic nutrition involves 5 distinct stages that work together to process food. Stage 1 is ingestion, where food is taken in through the mouth or specialized feeding structures. Stage 2 is digestion, where complex food molecules are broken down into simpler, absorbable nutrients through mechanical and chemical processes. Stage 3 is absorption, where digested nutrients move from the digestive tract into the bloodstream or body fluids. Stage 4 is assimilation, where the absorbed nutrients are used for energy production, growth, and cell repair. Stage 5 is egestion, where undigested and waste materials are eliminated from the body. Let's look at some examples of animals that use holozoic nutrition. Humans have an omnivorous digestive system with a long digestive tract that can process both plant and animal material. Dogs have a primarily carnivorous digestive system with a shorter intestinal tract optimized for digesting meat. Birds have specialized digestive adaptations, including a crop for food storage and a muscular gizzard for grinding food items. Let's review some key points about holozoic nutrition. Digestive systems vary greatly among organisms based on diet and evolutionary adaptations. All 5 stages, ingestion, digestion, absorption, assimilation, and egestion, are essential for complete nutrient extraction. Different animals have specialized digestive structures based on whether they're herbivores, carnivores, or omnivores. Holozoic nutrition is found in most animals, many protozoans, and some fungi. Saprophytic nutrition is a fascinating feeding strategy used by many organisms in nature. Saprophytes are organisms that feed on dead or decaying organic matter through a process of external digestion. Unlike other organisms, saprophytes don't ingest their food. Instead, they live directly on or near the decaying organic matter. They secrete powerful digestive enzymes that break down complex organic compounds externally. These enzymes break down complex organic compounds into simpler, absorbable forms like sugars and amino acids. Common examples of saprophytes include mushrooms and other fungi, many types of bacteria, and molds. Saprophytes play a crucial ecological role as decomposers in ecosystems. They break down dead organic matter, releasing nutrients that would otherwise remain locked in dead tissues. This recycling of nutrients is essential for soil formation and fertility. Without saprophytes, nutrients would remain trapped in dead organisms, and new plant growth would be limited. By breaking down organic matter, saprophytes serve as nature's recyclers, making them essential components of all ecosystems. Parasitic nutrition represents a specialized feeding strategy in the natural world. In this relationship, the parasite relies on its host for survival, extracting essential nutrients directly from the host organism. Parasites are categorized into 2 main types based on where they live in relation to their host. Ectoparasites live on the external surface of the host, attaching to skin, fur, or feathers to extract nutrients through the surface. Endoparasites live inside the host's body, inhabiting organs, blood vessels, or tissues, where they absorb nutrients directly from within. Let's examine some common examples of parasites and how they obtain nutrients from their hosts. Ticks are ectoparasites that attach to the skin of mammals with specialized mouthparts to feed on blood, often transmitting diseases in the process. Tapeworms are endoparasites that live in the intestines of vertebrates, where they absorb nutrients that the host has already digested. Some species can grow to several meters in length. Mistletoe is a parasitic plant that grows on the branches of trees, extracting water and minerals from its host while still performing some photosynthesis on its own. Parasites have evolved specialized mechanisms to extract nutrients from their hosts. Many parasites use physical attachment structures like hooks, suckers, or specialized mouthparts to penetrate host tissues and gain direct access to blood or bodily fluids. Some parasites, like tapeworms, have no digestive system and instead absorb nutrients directly through their body surface, taking pre-digested nutrients from the host's intestine. Many parasites secrete enzymes or chemicals that manipulate the host's physiology to divert nutrients to the parasite, essentially hijacking the host's systems for their own benefit. Despite their negative effects on individual hosts, parasites play important ecological roles in natural ecosystems. They help regulate host populations, influence community structure, increase biodiversity, drive evolutionary adaptations, and even serve as indicators of ecosystem health. In summary, parasitic nutrition represents a sophisticated feeding strategy where organisms extract nutrients from living hosts. This relationship ranges from relatively benign to severely harmful, with parasites evolving specialized adaptations to exploit their hosts, while often avoiding detection by the host's immune system. Herbivores are heterotrophs that obtain energy and nutrients exclusively from plant materials. They occupy a critical position in food chains as primary consumers, directly connecting plant producers to higher-level consumers. Herbivores have evolved specialized adaptations to efficiently process plant material, which is often difficult to digest. Most herbivores have specialized flat teeth with wide surfaces for grinding tough plant material. These teeth help break down cell walls and increase the surface area for digestive enzymes. Herbivores typically have longer and more complex digestive systems than carnivores. Ruminants like cows have multi-chambered stomachs that allow for fermentation and extended digestion of plant matter. Many herbivores rely on symbiotic relationships with microorganisms in their digestive tracts. These microbes produce enzymes that break down cellulose, a major component of plant cell walls that most animals cannot digest on their own. Common examples of herbivores include cows, which are ruminants with four-chambered stomachs, rabbits, which use hindgut fermentation, and elephants, which process massive quantities of plant material daily. Ecologically, herbivores play a crucial role as primary consumers. They convert plant energy into animal biomass, making it available to higher trophic levels in the ecosystem. Herbivores also help control plant populations through grazing, maintain ecosystem balance, and provide a critical food source for carnivores and omnivores. Carnivores are a specialized group of heterotrophs that primarily consume other animals for their nutrition. Carnivores have evolved specialized adaptations for their meat-eating lifestyle. These include sharp teeth for tearing flesh and powerful jaws for capturing prey. Unlike herbivores, carnivores have shorter digestive systems as animal tissues are easier to break down than plant material. This allows for more efficient nutrient absorption. They also possess enhanced sensory abilities and specialized hunting strategies that have evolved over time to make them effective predators. Carnivores exist in diverse forms across various habitats. Land carnivores like lions use pack hunting strategies, while eagles are adapted for aerial hunting with keen eyesight and strong talons. Aquatic carnivores, such as sharks, have specialized adaptations like streamlined bodies and sensitive electroreceptors to detect prey in water. In food chains, carnivores occupy important positions as secondary or tertiary consumers. Energy flows from producers through herbivores to various levels of carnivores. Carnivores play crucial ecological roles by regulating prey populations and preventing overgrazing by herbivores. They maintain ecosystem biodiversity by controlling species distribution and remove sick or weak individuals, improving the genetic health of prey populations. Carnivores are essential components of healthy ecosystems, balancing populations and maintaining biodiversity through their predatory activities. Their specialized adaptations reflect millions of years of evolution toward efficient predation strategies. Detritivores are essential heterotrophs that play a crucial role in recycling nutrients in ecosystems. Detritivores are organisms that feed on dead organic matter, also known as detritus. This includes fallen leaves, dead animals, and other organic waste materials. Unlike decomposers, which break down dead matter externally using enzymes, detritivores physically consume and digest detritus. Both groups are vital components in nutrient recycling. Common examples of detritivores include earthworms, millipedes, and various crustaceans like crabs. Detritivores play a critical role in nutrient cycling by converting dead organic matter back into usable nutrients that can be absorbed by plants. They form a vital link in the nutrient cycle. Plants are consumed by animals, which eventually die and become detritus. Detritivores then process this material, returning nutrients to the soil for plants to use again. Detritivores provide numerous benefits to ecosystems. They accelerate decomposition, improve soil quality through aeration, support plant growth by releasing nutrients, and prevent the accumulation of waste materials. As nature's recyclers, detritivores ensure the continuous flow of nutrients in ecosystems, demonstrating yet another fascinating adaptation among heterotrophic organisms. Organotrophs are a specialized type of heterotroph that obtain their electrons and energy from carbon-based organic compounds. Organotrophs break down various types of organic molecules, including carbohydrates, proteins, and lipids, to obtain energy. The primary process organotrophs use is cellular respiration. Here, glucose combines with oxygen to produce energy in the form of ATP molecules, while releasing carbon dioxide and water as waste products. Most animals, fungi, and many bacteria are organotrophs. These organisms rely on breaking down organic compounds from their environment or from other organisms. Let's examine how organotrophs process organic molecules. First, they consume food containing organic compounds. Through digestion, these complex molecules are broken down into simpler forms. Once absorbed into cells, these simple molecules undergo cellular respiration, generating ATP energy that powers all cellular activities. Lithotrophs are specialized organisms that obtain electrons from inorganic compounds for energy. The term lithotroph comes from the Greek words litho, meaning stone, and troph, meaning feeder. Lithotrophs can be categorized into two main types, autotrophic and heterotrophic. Autotrophic lithotrophs fix carbon dioxide and create their own organic compounds, making them completely independent of organic sources. In contrast, heterotrophic lithotrophs cannot fix carbon dioxide and require organic carbon sources, although they still use inorganic compounds for energy. The key difference is that both use inorganic compounds for electrons, but they differ in their carbon source. Let's examine some examples of heterotrophic lithotrophs, starting with iron-oxidizing and sulfur-oxidizing bacteria. Iron-oxidizing bacteria obtain energy by oxidizing ferrous iron to ferric iron. Similarly, sulfur-oxidizing bacteria derive energy from the oxidation of hydrogen sulfide or elemental sulfur to sulfate. Let's explore the simplified metabolic pathway of heterotrophic lithotrophs. The process begins with an inorganic compound, which serves as an electron donor. Electrons are extracted from this compound and fed into the electron transport chain. The electron transport chain generates energy in the form of ATP, which powers cellular processes. Unlike autotrophs, heterotrophic lithotrophs cannot fix carbon dioxide. They require organic carbon sources for biosynthesis. This is the key distinction. Heterotrophic lithotrophs obtain energy from inorganic compounds but require organic carbon for building cellular components. Heterotrophic lithotrophs have significant ecological importance. They contribute to biogeochemical cycling, thrive in extreme environments like hot springs and deep sea vents, and play important roles in metal corrosion and bioremediation. These remarkable organisms bridge the gap between the inorganic world and organic life, demonstrating the incredible diversity of metabolic strategies in nature. Fungi represent a major kingdom of heterotrophic organisms that cannot produce their own food. Instead, they obtain nutrients from organic materials, primarily through saprophytic nutrition. Fungi use a unique external digestion process. They secrete powerful digestive enzymes onto organic matter such as dead plants or animals. These enzymes break down complex compounds into simpler forms that can be absorbed through their hyphae. There are several major types of fungi. Mushrooms have visible fruiting bodies and complex underground networks. Molds grow as fuzzy colonies on organic matter like bread or fruit. Yeasts are single-celled fungi crucial for processes like baking and brewing. Fungi play a critical role as decomposers in ecosystems. They break down dead organic matter from plants and animals, releasing nutrients back into the soil. This recycling of nutrients is essential for ecosystem health and productivity. Without fungi, dead material would accumulate and nutrients would remain locked away. Many fungi form symbiotic relationships with plants called mycorrhizae. In these partnerships, fungal networks surround or penetrate plant roots, essentially extending the plant's root system. The fungi provide minerals and water to the plant, while receiving carbohydrates in return. Approximately 90% of land plants benefit from these fungal associations. To summarize, fungi are essential heterotrophs that obtain energy through saprophytic nutrition using external digestion via secreted enzymes. They exist in various forms, including mushrooms, molds, and yeasts. Ecologically, they function as crucial decomposers and form beneficial symbiotic relationships with many plants. Heterotrophic bacteria are microorganisms that obtain carbon from organic compounds, making them fundamentally different from autotrophs. Heterotrophic bacteria can be classified into three major categories: saprophytic, pathogenic, and symbiotic bacteria. Saprophytic bacteria decompose dead organic matter, recycling nutrients in the soil by releasing enzymes externally. Pathogenic bacteria cause diseases by obtaining nutrients from their host, often developing specialized adaptations. Symbiotic bacteria live in animal digestive tracts, aiding in digestion in a mutually beneficial relationship. Heterotrophic bacteria employ various nutritional strategies. They break down organic matter using enzymes, then absorb the resulting nutrients to generate energy, producing waste byproducts. Heterotrophic bacteria utilize three main metabolic pathways, depending on their environment. Aerobic respiration uses oxygen as the final electron acceptor, producing more ATP than other pathways. Anaerobic respiration uses alternate electron acceptors like nitrate or sulfate when oxygen isn't available. Fermentation occurs without external electron acceptors, typically yielding less energy but allowing bacteria to survive in oxygen-free environments. Let's examine some notable examples of heterotrophic bacteria. Escherichia coli, or E. coli, is found in intestines. While most strains are harmless, some can cause illness. It's a facultative anaerobe, meaning it can grow with or without oxygen. Clostridium species are strict anaerobes that form protective endospores. Some species cause serious diseases like tetanus and botulism, while others are found in soil and gut microbiomes. Lactobacillus produces lactic acid through fermentation. These bacteria offer probiotic benefits and are used in yogurt and other fermented food production. Heterotrophic bacteria play crucial roles in both ecology and medicine. Ecologically, these bacteria are vital for decomposition and nutrient cycling. They help fix nitrogen in soil, support plant growth, and maintain soil structure. In medicine, some heterotrophic bacteria cause infectious diseases, while others produce antibiotics and enzymes used in healthcare. They support digestive health, but can also be targets of antibiotic treatments. Photoheterotrophic bacteria represent a fascinating intersection between heterotrophs and autotrophs. These unique microorganisms use light for energy, similar to plants, but cannot fix carbon dioxide, requiring organic carbon sources like other heterotrophs. They exhibit several key characteristics that distinguish them from other bacteria. They harness light energy through phototrophy, but unlike autotrophs, they cannot fix carbon dioxide. Instead, they require organic carbon compounds for growth, demonstrating their heterotrophic nature. This dual metabolic approach is made possible by specialized pigments called bacteriochlorophylls. Bacteriochlorophylls are specialized pigment molecules that enable these bacteria to capture light energy. Unlike plant chlorophylls, bacteriochlorophylls can absorb light in the near-infrared spectrum and function in anaerobic conditions. The photosystem of these bacteria consists of a light-harvesting complex that captures photons and transfers the energy to a reaction center. This generates a flow of electrons and establishes a proton gradient that drives ATP synthesis. Two major groups of photoheterotrophic bacteria are the purple non-sulfur bacteria and green non-sulfur bacteria. Purple non-sulfur bacteria, like Rhodobacter and Rhodopseudomonas, can use various organic compounds such as acetate. They can grow in anaerobic conditions with light or aerobically in darkness. Green non-sulfur bacteria, including Chloroflexus and Acylochlorus, metabolize organic acids and sugars and are commonly found in hot springs and microbial mats. To understand where photoheterotrophs fit in the microbial world, let's compare them with photoautotrophs. Both use light as their energy source, but they differ in their carbon source. While photoautotrophs can fix carbon dioxide from the atmosphere, photoheterotrophs must obtain organic carbon compounds from their environment. Photoheterotrophic bacteria demonstrate nature's remarkable metabolic diversity. Their ability to use light for energy while requiring organic carbon illustrates the fascinating evolutionary adaptations of microorganisms to various ecological niches. Iron-reducing bacteria are specialized heterotrophs that use iron compounds in their metabolic processes. These bacteria reduce ferric iron Fe(Smus) to ferrous iron Fe(Swertmis) while oxidizing organic compounds. This process allows them to gain energy for their metabolism. Two important examples of iron-reducing bacteria are Geobacter and Shewanella. Geobacter is commonly found in anaerobic sediments and can transfer electrons directly through conductive appendages. Shewanella is found in aquatic environments and uses protein nanowires for electron transfer. Iron-reducing bacteria play a critical role in global iron cycling. They influence the availability of nutrients and can impact the mobility of environmental contaminants through their metabolic activities. Iron-reducing bacteria have promising applications in bioremediation. They can help clean up metal-contaminated sites by transforming toxic metals into less harmful forms. They're also used in treating industrial waste and even in generating electricity through microbial fuel cells. Animals represent the most familiar examples of heterotrophic organisms in our world. As obligate heterotrophs, animals must consume organic matter to obtain energy and nutrients for survival. Animals have evolved a remarkable diversity of feeding strategies to obtain nutrients. These include filter feeders that strain tiny food particles from water, herbivores that consume plant material, carnivores that hunt other animals, parasites that feed on host organisms, and detritivores that consume decaying matter. Animals have evolved specialized structures for obtaining and processing food. Teeth are specialized adaptations seen across different animal groups. Beaks are specialized adaptations seen across different animal groups. Filter systems are specialized adaptations seen across different animal groups. The animal kingdom represents an evolutionary spectrum of heterotrophic strategies, from simple to complex. This ranges from simple filter-feeding sponges to jellyfish with basic predation, to insects with diverse methods, to specialized fish, and finally to mammals with complex feeding adaptations. Within food webs, animals occupy different trophic levels as heterotrophs. Plants form the base as producers, while herbivorous animals consume plants, and carnivorous animals consume other animals. In conclusion, Animals exhibit remarkable diversity in their heterotrophic strategies, reflecting evolutionary adaptations to different ecological niches. Parasitic plants are unusual heterotrophs in the plant kingdom that have evolved to extract nutrients from other plants. Unlike typical plants that photosynthesize, these heterotrophs extract water, minerals, and organic compounds directly from their host plants. Through evolution, parasitic plants have reduced or completely lost their photosynthetic capabilities. They've developed specialized feeding structures, while some species maintain partial photosynthesis. The key adaptation in parasitic plants is the haustorium, a specialized feeding structure that penetrates the host's tissues to extract nutrients. Common examples include dodder, which appears as yellow or orange thread-like stems wrapping around host plants, mistletoe growing on tree branches, and broomrape emerging from host roots. Looking closely at how parasitic plants connect to their hosts, the haustorium penetrates through the host tissue to reach the vascular system, tapping into both the xylem and phloem to extract water, minerals, and carbohydrates. Parasitic plants can be classified based on their dependence on hosts. Holoparasites are completely dependent, while hemiparasites maintain some photosynthetic activity. They can also be obligate or facultative parasites, depending on whether they can survive without a host. Saprophytic plants are a fascinating group of heterotrophs in the plant kingdom. Unlike most plants, saprophytic plants have lost their chlorophyll and cannot photosynthesize. Instead, they obtain nutrients from decaying organic matter in the forest floor. These unusual plants evolved from autotrophic ancestors, adapting to life in low-light forest environments by developing specialized relationships with fungi. Their nutritional mechanism is particularly interesting. Saprophytic plants form myco-heterotrophic relationships with fungi. These fungi connect to tree roots, forming mycorrhizal networks, allowing the plants to effectively steal nutrients that the fungi obtain from trees. The Indian pipe, scientifically known as Monotropa uniflora, is a classic example. It has a distinctive pure white, waxy appearance due to its complete lack of chlorophyll, and is typically found in dense, shady forests. Another example is the ghost plant, or Monotropa hypophytis, which exhibits a yellowish or pinkish coloration. Unlike the Indian pipe, it produces multiple flowers on a single stem and is often found in coniferous forests. These specialized plants play unique roles in forest ecosystems. Saprophytic plants inhabit specific forest niches and can serve as indicators of mycorrhizal fungal health. They form part of the complex nutrient networks that exist beneath the forest floor. In the forest ecosystem, trees provide carbon to mycorrhizal fungi through their roots. The saprophytic plants then tap into this fungal network to extract the nutrients they need without directly contributing to the photosynthetic process. Through this specialized nutritional strategy, saprophytic plants have carved out a unique niche in forest ecosystems, demonstrating the remarkable diversity of heterotrophic adaptations in the plant kingdom. Heterotrophs occupy specific positions within food chains, forming the backbone of energy transfer in ecosystems. Food chains consist of multiple trophic levels, each representing a different role in the energy transfer system. Here's a classic food chain example. Grass is eaten by grasshoppers, which are eaten by frogs, then snakes, and finally hawks at the top of the chain. Energy transfer between trophic levels follows the 10% rule. When energy moves from one trophic level to the next, approximately 90% is lost as heat or through metabolic processes. Only 10% becomes incorporated into the biomass of the next level. This is why food chains rarely have more than 4 to 5 levels. There simply isn't enough energy left to support additional trophic levels. Let's explore how food chains vary across different ecosystems. Marine ecosystems often have longer food chains, starting with phytoplankton and ending with apex predators like sharks. Forest ecosystems might include food chains from oak leaves to caterpillars to songbirds and finally to foxes. Savanna ecosystems typically have shorter food chains, such as grass to zebras to lions, with fewer trophic levels overall. Heterotrophs form the majority of these trophic levels, with only producers at the base being autotrophs. Each ecosystem's food chain reflects the unique energy relationships between its organisms. Energy transfer in heterotrophs follows specific processes that determine how organisms obtain and use energy. Heterotrophs transfer energy through four main processes: ingestion, where food is taken in; digestion, where it's broken down; absorption, where nutrients enter the cells; and cellular respiration, where energy is extracted. Energy transfer between trophic levels is remarkably inefficient, with only about 10% of energy successfully passing from one level to the next. This inefficiency occurs for several reasons. Much energy is lost as heat during metabolism, used for movement, directed to bodily functions, and some biomass is never consumed. At the cellular level, energy is extracted through cellular respiration. When glucose is broken down, only about 40% of its energy is captured as ATP, the cell's energy currency. The remaining energy is lost as heat and used to form waste products like carbon dioxide, illustrating why energy transfer can never be 100% efficient. Terrestrial ecosystems host a diverse array of heterotrophs occupying various ecological niches. These organisms range from microscopic soil decomposers to large mammals, each with specialized feeding strategies adapted to their environments. Forest ecosystems feature multiple layers of heterotrophs. In the soil, microbes and fungi break down leaf litter. The forest floor hosts earthworms and beetles. Above ground, herbivores like deer feed on plants, while carnivores like wolves hunt other animals. Grassland ecosystems are dominated by grazers such as bison and grasshoppers that consume grasses. Predators like lions and eagles hunt these grazers. Underground, burrowers like prairie dogs create complex tunnel systems, while decomposers recycle nutrients. Desert heterotrophs have evolved specialized adaptations for water conservation and heat avoidance. Many are nocturnal, like kangaroo rats and scorpions. Predators, such as snakes, have highly efficient metabolism. Opportunistic omnivores, like coyotes, can utilize diverse food sources. Terrestrial heterotrophs have developed numerous adaptations to survive in their environments. These include water conservation strategies like specialized kidneys and nocturnal behavior, temperature regulation through hibernation or migration, feeding specializations with evolved mouthparts and hunting techniques, and seasonal adaptations such as food storage and fat accumulation. Terrestrial food webs illustrate the energy flow between heterotrophs. Plants produce energy through photosynthesis, which is consumed by herbivores like rabbits and insects. These primary consumers are eaten by secondary consumers such as foxes and hawks. Decomposers like fungi and bacteria break down organic matter from all trophic levels, returning nutrients to the soil for plants. Terrestrial heterotrophs demonstrate remarkable adaptations to diverse environments. From microscopic soil decomposers to apex predators, these organisms maintain ecosystem balance through their specialized feeding strategies and ecological roles, driving nutrient cycling throughout terrestrial biomes. Heterotrophs play critical roles in maintaining ecological balance within ecosystems. Predator-prey relationships are fundamental to ecological balance. When prey populations increase, predator populations follow, creating a natural balancing effect. Trophic cascades occur when changes at one level of a food web ripple down through other levels. For example, when wolves are removed from an ecosystem, deer populations can explode, leading to overgrazing of plants. Keystone species are heterotrophs that have a disproportionately large impact on their ecosystem. Sea otters are a classic example. They control sea urchin populations, which in turn prevents the destruction of kelp forests. When non-native heterotrophs are introduced to ecosystems, they can severely disrupt ecological balance. In Australia, cane toads poison native predators, leading to population explosions of the prey species these predators would normally control. Heterotrophs play a crucial role in nutrient cycling. As they consume organic matter and excrete waste, they make essential elements available for producers. Decomposers like fungi and bacteria complete this cycle by breaking down dead organisms.